Good afternoon, and thank you to the organizers for the opportunity to present on REGENXBIO. We at REGENXBIO have a mission to improve lives through the curative potential of gene therapy, and we do this by utilizing our proprietary set of AAV vectors from our NAV platform. And importantly, these include AAV8 and AAV9, which are the most clinically validated AAV vectors in the field across indications throughout the body. And this is in terms of safety, efficacy, and importantly, durability. Here you see our product profile, which includes 3 late-stage programs, 2 in our rare disease franchise, and also, of course, our 314 program, also known as Surovec, which we're developing for wet AMD and also treatment of diabetic retinopathy with our global partner, AbbVie. And we'll focus on Surovec for the rest of the presentation. So Surovec utilizes the AAV vector that I just mentioned to deliver a gene that encodes and produces the protein that is an anti-VEGF monoclonal antibody fragment with the goal to achieve sustained protein expression by the patient's own retinal cells. Now we're looking at 2 different routes of administration, both subretinal delivery as well as in-office suprachoroidal delivery. We initially focused on and continue to develop subretinal delivery, and we chose this because that is the most validated approach for safely and effectively delivering gene therapy to a retina target tissue. But we also felt it was important to follow on with an additional route of administration that would allow for in-office delivery. And we selected suprachoroidal because it preserved the positive aspects of subretinal delivery that were important to us. One is local delivery at the site of the target tissue, and also compartmentalized delivery to really decrease the exposure to off-target sites such as vitreous and anterior segment. This slide shows the various studies that we have with these 2 routes of administration. The most advanced is our subretinal delivery for treatment of wet AMD, which is in pivotal development globally with our Atmosphere and Ascent studies that are enrolling 1,200 patients globally. So the largest gene therapy trials ever conducted. And fortunately, we're advancing very well with these trials, and we're gonna have completion of enrollment to allow for top-line 1-year pivotal endpoint results readout next year. In addition to subretinal, we also have 2 suprachoroidal studies ongoing. One is in AAV8 for treatment of wet AMD as well, and also Altitude for treatment of diabetic retinopathy. And I'm going to spend the rest of the talk speaking about diabetic retinopathy and the Altitude study. We all know that diabetic retinopathy is a global health problem. We also know that in clinical trial settings under tight control, that existing approved anti-VEGF agents, when given repeatedly, can treat this disease and prevent progression to vision-threatening complications. Unfortunately, we also know that in the real world, patients are not getting this treatment because of the difficulties and the treatment burden with repeat visits and injections. So patients are getting worse. So herein lies the opportunity for gene therapy to potentially have a one-time in-office treatment that can potentially stop the clock on DR progression and ultimately lead to decreased vision-threatening complications. So here is our DR study, which is enrolling patients with moderately severe NPDR, severe NPDR, and also mild PDR. And we've enrolled across 3 ascending cohort patient populations. And what I'm going to present today is the 1-year results with the first 2 dose levels. Baseline characteristics have been well matched. Starting with safety, Surovec has been well tolerated. An important point is with dose levels 1 and 2, there was no prophylactic steroid regimen used. We saw a low rate of intraocular inflammation, where importantly, all these events were mild and resolved with very short course topical prophylaxis. On to efficacy. So we included a control arm of no treatment, and not surprisingly, we see that these patients generally do worse, even within a year. And in fact, 38% of patients had at least two-step worsening. In contrast, Surovec at dose level 1 and dose level 2, we saw patients generally stable to improve, and in dose level 2, 100% of the patients were stable to improved. Critically, we're seeing these imaging results translate into benefit in terms of vision-threatening complications. So we see a dose-dependent reduction with notably an 89% reduction in vision-threatening complications with dose level 2 in NPDR patients. Here we have 3 cases from one of the investigators, Arshad Kanani, and in the interest in time, I'm not going to go through each of these, but you can see a couple cases here representative of what we saw in the trial, where patients treated with dose level 2 have stable to improved disease severity, while a control patient that was treated had significant worsening. So in conclusion, Surovec has been well tolerated, and we're seeing very encouraging efficacy signals in our phase 2 study, including stability and improvement on DRSS and a dramatic reduction in VTE rates. So based on these encouraging results, we in AbbVie are excited about advancing plans for a pivotal global program for diabetic retinopathy. And we look forward to sharing more updates on this program going forward. Thank you.